Okay. Can everyone see? Clear? Okay, I'm gonna present data that I've been working on for the past year with my advisor Ricardo Gonzalez Pinzon and other collaborators from SWell. And I'm gonna be presenting on biogeochemical constraints of nutrient processing along river corridors. And part of the motivation for this study has to do with, in part, the current status quo of our field and the impairments of rivers across the United States. So of all the rivers in the United States, there are about 31% of them that are accessible. And of those accessible rivers, we have about 52 of them that are impaired. Those impairments are usually based because of some pathogenic sources of sediments, transport of nutrients. And nutrients is what I want to focus on here today. And when I say nutrients, I'm talking about nitrogen, carbon, and phosphorus, so I'm going to limit it to that. And the main source of those nutrients are mostly from agriculture activities. And so that's the problem. And these impairments cost billions of dollars every year in terms of like remediation and everything to US taxpayer. And so there's a very important need to understand these processes from a mechanistic view. And now the status quo now, why I have my title as river corridors. If you look over all of the research papers that are available in like nutrients impairment across our field of hydrology, you see that a lot of the research that have been done in first order streams. And now like higher order streams, there's not a lot of research. And what this does is that it creates a lack in the transferability of knowledge from first order systems to higher order system. And so there is a very, very strong need to sort of couple these two systems together and not to treat them as separate systems, so that we can understand better how nutrients are processed along an entire river corridor. And so for this process, we developed three objectives. The first objective is to investigate how changes in sediment texture across resource supply and colonization area of microbial communities affect nutrients processing along the entire continuum. Second, we looked at investigating the differences in microbial diversity, community structure, and genomic potential in the sense of how much does evolution of these microbial communities and stoichiometry might affect how much nutrients that we see, nutrients uptake we see across the continuum. The last one was to investigate the effect of dissolved organic matter. And the effect of dissolved organic matter, that is usually prevalent in like low order stream, like first order stream, compared to mid order stream, and it's also higher in some higher order stream. And so for this, we developed sets of hypotheses, which I'm going to just give in terms of like a diagram. So we hypothesized first that we're going to see higher nutrients uptake. Nutrients uptake is going to decrease with increase in nitrogen concentration. And so if you look at like A1 here, the x-axis, that is nitrate nutrients concentration, and the y-axis VF here is the uptake of nutrients. And here we're saying that because DOC quality is so high, we expect that the rest-free ratio, which I'm going to talk about later, when we use the rest-free ratio, we should see higher uptake in those regions, and compared to just using like nitrogen only injection, where we might see some sort of P or C limitations in terms of like it reducing like the uptake of nutrients that we see in our systems. And we also go about mid-order streams where we have like lower DOC, where we might even see more decrease in nutrients uptake because, again, of that limitation in carbon and also like higher increase in nitrogen and phosphorus. And I also created a diagram where we expect that across the entire continuum to see a steady increase and a steady decrease in nutrients uptake as we move along the continuum. So to test this hypothesis, we conduct sets of experiments across the river continuum. And as you can see, we did a first order all the way down to 7th order B. We're not able to get to 8th order. And so these experiments included like the incubation of column. As you see here, like PVC pipes, we incubated these for about a month. And after incubation, we brought these back to the lab and we injected conservative tracers and reactive tracers to them. And for these injections, we did 2 sets of injections: the nitrate-only injection and the RESV injection. And the RESV injection has been sort of like agreed upon, or like there was a very beautiful paper by RESV back in I think like the 30s or 20s, where he found that 106, 16, or 1 are the ideal ratios to get optimum uptake, nutrients uptake. It was not in like a stream ecosystem, but it was more for like ocean, like saltwater systems. But we use the same relationship. And so I'm going to show a couple of results, and on those results, you're going to see nitrate and you're going to see redfield. So you understand that those were 2 sets of injections that we did. And so for these injections,
6 minutes, Jancoba.
For these injections, so the first thing we did was like use reactive models, and first we have to fit them into breakthrough curves. And then from those breakthrough curves, we're able to estimate up to parameters across the breakthrough curve in nitrate and redfield, and try to see how they scale. And we're able to create a correlation across the sites for redfield and across sediment texture. And from that, we concluded that variation in transport conditions induced by differences in stream and sediment characteristics directly influence nutrients uptake and may have ecological implications. Next, we say it is possible to achieve knowledge transferability. For our future work, we're going to be working with folks from PNNL to try to understand, like, try to do DNA sequencing to the microbial community to understand how these genomic types and functions might have affected nutrients uptake. And also the development of a mechanistic model where we can use to scale nutrients processing across the river continuum. For that, thanks, and thanks to all of the organization and CWE for support.
Thank you, Jancoba. Does anybody have any questions for Jancoba?
I have a quick question, because this is the 3rd presentation in a row where you've talked about ordered streams. What the hell is that? I mean, what's the difference between a 1st, a 3rd, and a 9th?
So what happened is that your first order stream, you think of that as your source water, right? Like the Martinez stream, that's where like the entire river starts, right? And then as you come, so for instance, like there's a hierarchical classification of how they are. So you have a first order stream, and then you have a second order stream only if you have 2 first order streams connecting together. And you have a third order stream only if you have 2 second order streams connecting together, and back and so on and so forth. And so with that comes, like, changes in like stoichiometric conditions in these systems, right? And so that's the importance of those orders.
Thank you. I thought it was something like that. I had it backwards, but yeah, thanks for that.
Yeah.
I had a question. Were you doing the acetate and nitrogen and phosphorus additions in situ or in the lab? I didn't quite catch that.
I was doing that in the lab. So we decided to do it in the lab because we can have more control compared to doing, like, usually we do like nutrients injection in the stream, but in the lab we have more control of our parameters.
I'm curious, so you do them sometimes in the stream. If you do that, do you need a special permit? Because you're really adding a pollutant into the stream.
Yeah. We do need a special permit. We get special permit from I think Bureau of Rec. Yeah.
Thanks.
Hey, Jancoba, what is the, good job, by the way, you also a ton of data. What is the difference from stream orders in comparison? What's the temperature looking like for each, like for stream conditions, between something that's quite small and reactive to the environment to like the bigger body of water? Do you see a big difference between temperature for those order of streams?
We do see a big difference between temperature in those streams as well. Because I mean, it has to do with wave, and usually in like high order, like low order streams, it's Martinez, and so it's very cold up there, and so the like temperature changes, and it also affects pH and stuff like that. So we do see like drastic change in temperature, like water temperature. Yeah.
Do you have any two-parter? Do you have any insight into when, like out here in the out west in the mountains, those first order streams, we think of like those trout fishing streams up like in the mountains, but like say somewhere in the Midwest, you can have first order streams on basically the same elevation and they just keep dumping into each other and getting bigger. Is what you're doing, would that apply to the lowland stream orders as well, 1 through 9?
Actually, yeah, you're not going to the second chapter of my dissertation.
Oh,
You have to
great.
wait for that. Yeah, but yeah, we looked at that and we did a set of experiment across different systems in Iowa and New Mexico and Colorado, and we're looking at those kind of effects as well.
Cool. Great job. Thanks.
Yeah.
All right, thank you, Jancoba. Great presentation. All right, next up.